The study of the immune system is somewhat complicated, since its pedagogy consists of studying several separate phenomena that occur simultaneously in real life. It's like analyzing the pieces of a puzzle separately. Suppose that a person suffers a cut on the hand, therefore there is entry of bacteria beyond the skin, to the subcutaneous cellular tissue. At that moment, the first barrier of innate immunity is broken, which is the skin, which is a species of a wall like the one that exists in the kingdoms of antiquity. At that moment, inflammation occurs, with which there is an increase in local temperature and an increase in blood flow, with which there is a greater influx of defense cells to the site of injury. Bacteria have molecules called antigens on their surface, which are something like their fingerprint. Innate immune cells such as macrophages are capable of recognizing these antigens and their phagocytize. That is, they swallow this bacteria fragment, then inside, and then express these antigens of the bacteria, foreign to the organism on their own surface, using molecules called major histocompatibility complex molecules, HCM. Behaves something like a gossip neighbor in the neighborhood, and travels to the satellite ganglia, which are similar to train stations, where acquired immunity begins, where it presents these antigens to T lymphocytes. There are lymphocytes that present receptor TCR specific for a very wide variety of antigens, millions, and when the lymphocytes specific for these antigens meet the macrophage that presents the antigen, it is activated, proliferates, and differentiates to be even more precision. Memory T lymphocytes are generated, which remain circulating in the bloodstream, waiting for a possible second encounter with the pathogen, and effector T lymphocytes, collaborators, which thanks to chemotaxis go to the precise place where the infection is located, bacteria, and once they encounter it, they eliminate it. On the other hand, there are another type of lymphocytes, B lymphocytes, which have a receptor on their surface, known as BCR, which once they identify their specific antigen of the bacteria, they also activate, proliferate, and differentiate with the peculiarity that they give off their receptor, that is the BCR, to the serum and are known as antibodies or immunoglobulins, which have the ability to adhere to bacteria by anchoring to their antigens.
Así como los virus, los hongos, los parásitos, que son precisamente esos patógenos de los cuales nos defiende el sistema inmunitario, tienen en su superficie unas moléculas llamadas antígenos. ¿Bien?